Thanks a lot, Wolf. And sorry for the delay. I have 20 slides, so I hope we'll be able to manage in a decent amount of time. I think nobody in, I think nobody in this room needs to be convinced that our brain is the most complex, but also the most fascinating organ that nature has generated. Our brain contains 87 billion neurons that have to be born at the right time, migrate to the right position, and then wire up in the right way in order to allow us to perform all the cognitive processes that make us human. But unfortunately, this complexity comes at a price. Our brain is also the home of some of the most devastating human diseases, whether it's depression, epilepsy, schizophrenia, autism, dementia, or other neurodegenerative disorders like Parkinson. And it's currently thought that about 20% of the entire population at some point in their life will suffer from one of these diseases. Now, brain disorders are generally classified by their cognitive and by their behavioral symptoms. But what's underlying them is changes in neural network activity that are thought to be caused by altered architecture of neural networks. And what I would like to convince you of today is that we can use three-dimensional stem cell-derived cell cultures, so-called organoids, in order to study both the alterations in neural network activity and also changes in the underlying activity. About 10 years ago, Madeleine Lancaster, when she was in my lab, developed a methodology that at that time we called the cerebral organoid method. We take pluripotent stem cells, iPS cells, that we can derive from any patient, whether healthy or diseased, to grow these cerebral organoids, which are shown on the left-hand side of the slide. They are pretty unspectacular, untransparent balls of tissue, but when we section through them at an early stage of development, you can see that their cytoarchitecture, at least at the early stages of development, very much resembles that of a developing human brain. But most importantly, we can use these organoids to model neurodevelopmental disorders. So on top, you see a healthy, and on the bottom, a patient who suffers from severe microcephaly. We generated iPS cells from both healthy and microcephalic patients, grew organoids, and the organoids that were derived from the microcephaly patients were severely smaller. And this allowed us to identify not only the gene that was responsible for this microcephaly, but also the cellular mechanism that causes these brains to be much smaller. Now, this is all published, so I'm not going to go into details of this, but instead what I would like to talk about today is another disease that is called tuberous sclerosis, where we made some really, in my view, interesting and very surprising findings. Tuberous sclerosis is a very severe form of genetically caused epilepsy. It is characterized by two types of morphological alterations. On top, I show you the so-called cortical tubers that can be recognized by MRI and are actually inclusions and balls of neurons that are enlarged, so-called dysmorphic giant neurons, and they form in the cortex. And on the bottom, I show you benign tumors that protrude into the ventricles, and sometimes they obstruct the flow of cerebrospinal fluid that results in hydrocephalus and can be another severe symptom of those this disease. Tuberous sclerosis is caused by mutations in either TSC1 or TSC2. These are inhibitors of mTOR signaling. And based on many experiments that have been done in rodent models, it was thought that the patients have a heterozygous germline mutation in, for example, TSC2, and that at some point during the development, there is a spontaneous mutation that inactivates the second allele. And this is how the disease arises. This is based on mouse models. In mouse, if you're heterozygous for TSC2, you have absolutely no phenotype. You need to inactivate the second allele, and then you can recapitulate some of the features of tuberous sclerosis. And there has also been an organoid model where this was used to model some aspects of the disorder. But we didn't quite believe that. And when I say we, I'm talking about Oliver Eichmüller, a very talented medical student, now medical doctor in my lab, Nina Corsini, who is a research associate in my lab, and Marta Voigt, our clinical collaborator. I don't want to go into the details of our data. Basically, we recruited a cohort of patients, at that time 2, now 4 patients, generated iPS cells from them, always made an isogenic control. And I should mention at this point that none of the data points that I will show you throughout my talk, or all of the data points that I'm going to show you throughout my talk, are based on experiments with at least 2 patients plus their isogenic control. And every data point is 3 biological replicates, meaning it's 3 batches of organoids, not individual organoids. And what my lab found was that we can recapitulate the features of tuberous sclerosis. We can grow organoids to 130 days, and we can use a cell culture medium that is called BrainFizz, which supports the differentiation of neurons. And what we then find is shown on the middle panel. There are highly enlarged neurons that occur in statistically very high, highly significant numbers, and morphologically perfectly resemble the cortical tubers that form in tuberous sclerosis patients. We can also grow them in a different cell culture medium that contains high nutrients and promotes the proliferation of the neuroprogenitor cells in the organoids. And then we see, as you can see in the bottom part of the slide, the growth of these tumors that are highly positive for phospho-S6, indicating enhanced mTOR signaling, and are highly proliferative because they're positive for Ki67. So using different cell culture media, we can recapitulate the morphological features of tuberous sclerosis. Now we can ask an important question. Was it really the loss of the second allele that caused this disease to occur? For this, we simply dissected a few of these tumors and did a simple PCR reaction to determine the genotype at the TSC2 locus. And what you can see in this panel, where I show you the original sequencing traces, is that the vast majority of all the tumors were actually heterozygous for TSC2. There are a few, these are the largest tumors that are actually homozygous. We sequenced the genome of some of these, and what you can see, the red arrows indicate that, is that the tip of the left arm of chromosome 16 had become homozygous, or in other cases, the entire left arm of chromosome 16 had become homozygous. So there's 3 things that we can learn from this. The first one is it is not the loss of the second allele that is initiating the disease in humans. The second one is the loss of the second allele is a late consequence of tumor formation. And when it occurs by a phenomenon that's called copy-neutral loss of heterozygosity. And the third one is that we have to take all the literature on this disease that's based on rodent models with a grain of salt. And we hypothesized that there might be a cell type in the developing human brain that you do not find in mice that might be particularly sensitive to levels of mTOR signaling. And we identified this cell type doing numerous single-cell transcriptome experiments. I'm just going to show you one of them. On the left-hand side, you see a UMAP of a single-cell experiment where we have intermingled the cells from a control organoid in orange and the cells from a TSC mutant organoid in blue. And what you can see is that by and large, the different classes of neurons are equally represented in the 2 genotypes, but there are 2 clusters, one containing interneurons and the other one a new cell type that are massively overrepresented in the TSC mutant organoids. Based on the marker expression, we located these proliferating progenitor cells that are overrepresented in the TSC mutants to an area that is called the caudal ganglionic eminence. On the bottom right, I show you marker expression. One marker is called secretagoguin, the other one is the EGF receptor, and they're uniquely expressed at this stage of development in this area. What we also found by co-clustering the progenitor cells with cells from the actual human brain is that they match late stages of gestation, 21 or 22 weeks of gestation, or even later. And so we called them caudal late interneuron progenitors, or clip cells. And I would like to summarize a large part of work, which I don't go into details of because it was published last year. Basically, our work supported the following model, where in TSC, the overproliferation of these clip cells causes the formation of tumors in the caudal ganglionic eminence, which are overproliferating, and then some of these cells start generating interneurons, which then migrate into the cortex, where often they become too large and they form these tubers. In a healthy human fetus, the clip cells proliferate late in gestation, and they generate interneurons that then migrate into the cortex. But in order to differentiate into interneurons, mTOR signaling needs to be down-regulated. In TSC, in tuberous sclerosis patient organoids, this is not possible, and it's the upregulation of TSC, of mTOR signaling that causes the continued proliferation of those cells, and also that is responsible for the increase in the size of those cells. And again, this model is based on a huge number of experiments that are all published in this paper that I'm showing. Now, I told you in the beginning that tuberous sclerosis is actually a severe form of epilepsy. And so what I want to do during the rest of my talk is I would like to convince you that we can also model the neural network activity that underlies epileptogenesis in these organoids. We were motivated to do those experiments by the experiment that I show you here. This is a simple calcium imaging. We're using GCAM6S in organoids, and you see that the neural activity in an organoid is highly non-random. What you see is that there are groups of cells that are flickering, and then there is this massive burst of activity that that includes almost all of the neurons in an organoid. In the collaboration with Manuel Zimmer, a neuroscientist on our campus, we developed an algorithm that we can use to segment movies like this one and isolate each and every neuron in these movies. And what you can see in the traces that I show you in blue and red, in the bottom part of the slide, is that there's a certain frequency about every 30 seconds when the control organoids fire these bursts of activity, and the frequency of these activities is seriously increased in TSC mutant organoids. So this motivated us that we can model changes in neural network activity that underlie epileptogenesis in organoids. But the first thing, the disadvantage of calcium imaging, of course, is that it's very indirect and it is very slow. And so we asked, can we do electrophysiology in organoids? And for this, we used extracellular recording. We use silicon arrays, 4 shanks that contain 16 electrodes each. I show you a raw recording on the right-hand side, and you can see also this burst of activity that occurs in parallel with the GCAM burst that I showed you before. In these panels, we have thresholded the electrical activity, and on the left-hand side, I show you control organoid, and you can see that they occur very regular. On the right-hand side, I show you a TSC2 mutant organoid, and there's a number of things that you can see. The first one is the bursts are more frequent. The second one is that it's a higher fraction of all the neurons that participate. And the third one is, if I would focus into this, you could also see that the bursts are actually shorter in TSC2, but I'm going to come back to that later. Now, the great advantage of extracellular electrical recording over calcium imaging is that the signals are much richer. They contain much, much more information. And so we wanted to ask, can we use the information in this to estimate the degree to which we model the neural network activity changes that occur in TSC patients? For this, we first needed to know what are those changes. And for this, we collaborate with Maike Zeilmans at the Utrecht Medical Center. She's a neurosurgeon who performs surgery on TSC patients. And the way these surgeries are done is the skull is opened, and there are these beds of electrodes that are placed directly onto the open brain. And recordings from all these electrodes are processed in parallel. And they're processed before the surgery and after the surgery. And the recordings look a little bit like this one. So in an unprocessed signal, not very much can be seen. But if we put a bandpass filter of 80 to 250 hertz over it, there's characteristic differences between physiological and pathological high-frequency oscillations. In the physiological high-frequency oscillations, the amplitude is much lower, but the bursts are longer. Now, the way this is done is that the surgeon actually has a lot of experience with interpreting these signals, and when we talk with them, they tell us about the good ripples and the bad ripples. And so we wanted to ask, can we put a bit more quantitative estimates over this? And we found a number of parameters. First of all, the duration of the bursts is shorter in the pathological situation, but their amplitude is a lot higher, both the maximum amplitude and also the mean amplitude. And we can also calculate an entropy by integrating over different frequency ranges, and that's also significantly increased. So there's a set of features that distinguishes physiological versus pathological neural network activity. And so we wanted to know how much of this is recapitulated in organoid. This is a raw trace of a control and a TSC organoid. Just like in the extracellular recordings, we can put a bandpass filter and isolate the ripple band. Sorry, the ripple is the term that people doing these recordings are using for the frequency range between 80 and 250 hertz. For those of you who are in the field, there is also a fast ripple band, which is above 250 hertz. And we have the exact same data for this fast ripple band. I just don't want to confuse the audience with those details. If we look at the ripple band, we find that in the control, the oscillations are longer, and in TSC, they have a higher oscillation. So when we quantify this, in the bottom left, each dot represents one burst of activity. Duration is on the x-axis and the mean amplitude on the y-axis, and you can see that in general, the red ones are along the y-axis, meaning their amplitude is higher and the duration is shorter. The interburst interval is shorter in TSC, and again, the entropy is higher. So in summary, there's a number of features that distinguish physiological from pathological activity, which affect the duration, amplitude, frequency, power, and also entropy of these signals. And each and every one of them can be recapitulated in the organoid. And I should say that the quality of these recordings, of course, in organoids is much, much higher than what you can actually do during the surgery. But there has been reports that have indicated that there would be a very complex feature of physiological versus pathological ripple activity. And this affects the coupling of high-frequency oscillations to low-frequency oscillations. And I'm going to show you what I mean by this. So there's oscillatory activity in various different frequency ranges, and one of these ranges is the theta range. For those of you who are in the field, I would like to make it very clear that I'm not talking about what is physiologically called theta oscillations, including all the cognitive processes that are underlying this. What I'm talking about is simply oscillatory activity that is between 3 and 8 hertz. And it has been found that the physiological high-frequency oscillations occur when the theta oscillations are at their peak and start to move towards their trough. And when they move from trough to peak, these are more likely to be pathological high-frequency oscillations. So this is a very complex feature whose neural network correlate is a bit enigmatic. And we wanted to know, can we actually recapitulate this in an organoid? I show you these circular plots. This is typically how data like this are shown. I want you to focus on the left-hand side, and the blue part is the control activity. And you can see there's a strong peak of activity when the theta oscillation is just moving from trough to a peak. And in the TSC mutant organoids, there's a second peak that is developing, which represents the pathological high-frequency oscillations. And when we look at the amplitude of this, the amplitude is actually higher. And again, all of this is seen in multiple patients, and it's in multiple batches, and it's highly statistically significant. So what we conclude from this is that even very complex changes in neural network activity can be recapitulated in the tuberous sclerosis organoid model. And what we can do in this model now is we can ask, what's the underlying change in cellular activity that is actually responsible for this? For this, we put a bandpass filter of above 4,000 hertz over our data, and that allows us to isolate individual spikes, which correspond to individual action potentials. We did spike sorting, and we then performed principal component analysis, and the computer identified 3 types of spikes for us. The spike type 1 and the spike type 3 have an initial upward curve that is higher than the second one, indicating that they are excitatory, whereas the spike type 2 is predicted to be inhibitory. If we look at the distribution of these different types of spikes between control and TSC organoids, we find that it is the spike type 2, the inhibitory interneuron activity, which is much higher in the TSC organoid. What we can also do is we can pattern the organoids in a way so that they do not contain a caudal ganglionic eminence, and then these clip cells that I was telling you about in the beginning are not formed, and then you do not see this spike type 2. And when we look at the high frequency events, they are basically gone. Together, this suggests to us that it might be the neurons that are generated by these clip cells that are responsible for the pathogenic activity that we see in TSC. And to gain more evidence for this, we use the different protocols to pattern the organoids in a different way. And here we use a marker for the neurons from the clip cells that's called secreta boggin. In an unpatterned organoid, which I show you on the left-hand side, there's many of these neurons, but when we pattern them to dorsal, they're basically gone. And when we look at the ripple activity in the unpatterned, we find them in almost every organoid, but if we pattern them to dorsal, then this pathological activity is gone. So taken together, this data suggests to us that it is the neurons generated by these clip cells that are responsible for generating the pathological activity. And to get more evidence for this, we actually analyzed them cytologically. On the left-hand side, I show you a clip cell-derived neurons, a clip cell-derived neuron from a healthy control organoid, and you can see that these cells form very elaborate dendritic trees. On the right-hand side, I show you a similar cell from a TSC organoid, and what you can probably see is that there are these beads along the dendrites. This is a phenomenon that's called dendritic speeding that has been described before, but not in tuberous sclerosis, and is typically associated with hyperactivation of neuronal activity. And we find the same phenomenon when we analyze these neurons in actual sections from resected material from actual TSC patients. So taken together, these data suggest that the human brain contains a cell type that makes us more vulnerable to genetic alterations in mTOR signaling and ultimately to the development of epileptogenic activity. And there's one final question that remains here, which is, why does nature do this to us? Why do we have these cells? Now, there are certain phenomena that only occur in the human and in primate brains, but not in rodent brains. One of them has been identified in the lab of Arturo Alves Buella a couple of years ago, which is the so-called ARC. There is a massive stream of interneurons that migrates into the human prefrontal cortex after birth. It's very hard to analyze this phenomenon because they don't occur in typical laboratory animals. But Saku Wong, a very courageous graduate student in my lab, grew organoids up to 400 days. And what you can see, and what we have a lot of more evidence for that I don't have the time to show you, is that we can recapitulate this stream in organoids. And what these neurons are doing is they are expressing all the markers that we know are expressed in these neurons that come from the Klipp cells. And to gain more evidence for this, we performed histology and electron microscopy. So here I show you these ARC neurons in histology on the left-hand side and electron microscopy on the right-hand side. On the left-hand side, you see in blue these ARC neurons, and they're surrounded by a shell of astrocytes that you can also see on the right-hand side in electron microscopy. All of this was done in the lab of Jose Manuel Garcia Verdugo in Valencia. And so we teamed up with them and gave them samples of our organoids. And on the left-hand side, I show you the organoid analysis in histology, and the right-hand side, electron microscopy. And what they told us is that the cytology of this in the organoid is essentially indistinguishable from what they find in a real human brain. And so the final question that I wanted to ask is, how do these astrocytes, how does this come about? How do the neurons arrange themselves? And we're only at the beginning of understanding this, but Saku has generated an in vitro system where we grow these organoids in culture. Then we isolate those Klipp cells, grow them as neurospheres, and put them onto matrigel. And then we can generate movies like the one on the bottom left-hand side, where you can see that the neurons are starting to migrate out, and they're starting to arrange themselves into those streams. On the right-hand side, I show you immunofluorescence, and you can see that in the center of those streams are DLX56 positive interneurons, whereas on the periphery are these astrocytes. And so we hope that we will be able to study the mechanism of this cellular self-arrangement that is typical for this type of migration in vitro. So to summarize what I've shown you, I've told you that organoids can recapitulate tuberous sclerosis, both tubers and tumors, that we've identified the Klipp cells that are responsible for this, that the neural network activity changes in the TSC organoids in a way that's very similar to what's seen in TSC patients, and that the Klipp cells correlate with these neural network activity phenotypes, and that we can that our data indicate that the Klipp cells are responsible for this postnatal neural migration into the prefrontal cortex. And we hope that with this discovery, we will be able to eventually determine the role of this unusual phenomenon for the probably plasticity or the function of the human cortex, and why this actually was developed in higher animals, and why it's not necessary in mice. So in the end, I just wanted to acknowledge people. I mentioned them along the way. Oliver Eichmüller really deserves most of the credit for all of this. I also would like to acknowledge Michael Zabolotsky, who is a computational neuroscientist and has done much of the interpretation. And I would like to mention that this is a collaboration with Cedric Bardi at Summary in Adelaide. And I also would like to acknowledge our clinical collaborators, Michael Seilmans at the Medical Center in Utrecht, Mercedes Paredes at UCSF Department of Neurology, and Marta Feucht at the Vienna Medical University. And thanks for listening to me, and I'll be happy to take any questions.